Okay, so in this video we are going to discuss, reproduction in bacteria we were discussing, and that asexual which was done. And in today's video we are going to see like not true asexual, which simply means like no sexual, but in that we just consider we have three indirect steps. So what that first is conjugation, transformation, and transduction. We have these three steps. So we are starting with the conjugation first. First thing, this is like the process by which bacteria transfer their genetic material. So transfer of genetic material happen through this one, conjugation, transfer genetic material. We also said that the genetic material is transferred like by direct contact. Even you can understand direct contact in this picture, I will show you later on. So they have the direct contact transfer of the genetic material, and in this process you will see the DNA, and in this one you will see plasmid is transferred. So what is plasmid? So I could just make a quick picture. It's like simply a structure you will see. So here around like a structure you will see called as plasmid. So you will see in this DNA this plasmid would be transferred, and here you will see we have DNA, and so we are just saying that here we have chromosomal DNA, and you will see that this become donor. This means why? Because this plasmid is given, so they become pilus, and this is recipient, which means it will accept. And here we also have DNA. I'm sure this is not looking very good diagram, but I do have a picture where you can get it like very easily. Well, you just saw that plasmid will be transferred from one bacterium, which is like donor, transfer from donor, and then it will go to the matting to the recipient. This will go to recipient, which simply means the acceptor. And how will that travel? That will travel through pilus. So pilus is like, if this is plasmid, so plasmid will transfer like this structure, that's called as pilus. And the pilus or this pilus is also called as cytoplasmic bridge, because this is in the cytoplasm, and that connect one part to the one structure to another, cytoplasmic bridge. And you will see that they have the parasexual mode of nutrition. This means they're not completely sexual, parasexual like half sexual. So they have parasexual mode of nutrition. And what you will see more, and yes, here you will see that in bacterial conjugation the process, it's like only transfer of the genome. So genome is transferred, and you will see that a portion of the genome will be transferred. Plasmid from donor to recipient will be transferred through sexual reproduction, and you will see that undirectionally or like few other steps like separation of the cell will also occur. Separation of the cell, or that step is also called as recombination. So what is recombination? Recombination is simply a process where no population increase. I have just told in like last few videos as well. So population remains same, even not decrease, not even increase. This simply means if this the genome, so they only transfer, exchange of DNA will happen, but no population. This means they cannot make copy of itself. No copy of itself will be formed. That's called as recombination. So you will see there will be recombination of donor or like the genetic material to the recipient cell, which is the second one. And this one you will see that cell which will not completely considers asexual, like no matter how much sexual and asexual reproduction occur, but will consider parasexual or non-complete asexual reproduction, not complete sexual reproduction, because we cannot for sure consider them. Whereas I have few pictures I would love to show you. This is the picture I have taken from my book. Here you will see first is like, I cannot use pencil here, it's okay then. Okay, let's move. Like here you will see that they have the chromosomal DNA, and plasmid is the structure, pilus, which is the middle part present in the cytoplasmic, or this also called as cytoplasmic bridge. So you just saw this has been transferred, this is called donor, and the one who's accepting is called as recipient. I'm so tired. Well, here you will see that this plasmid will be transferred from pilus, through pilus to another recipient. When they got transfer, you will see the one part is going to be transferred here, and that have been entered to the second part. Here in like second step, you will see they become DNA polymerase will happen. Here it will be relaxosome transferosome. This means they got transfer of few things. Here this is new donor will be formed, because here all the steps are done. So you'll see that again this process would be continued. How continued in a sense, like here we have plasmid single, but when it's going to this one, you will see that here new plasmid will be formed. If new plasmid will be formed, so we must have pilus separately. So this is the new donor, here this is the old donor. So this is about the conjugation process. I'm sure it's clear to you. Okay, so thank you for watching. I will continue to more steps in upcoming video. Bye bye.